left off on after passive transport. Okay, so regarding to passive transport, concentration, yeah, Doug? Yeah, the reason why is because Alright, so regarding to passive transport, first of all, does it use ATP or not? No, right? Passive transport does not use ATP. Second of all, things always go from what kind of concentration to what kind of concentration? Always from high concentration to low concentration. So given this example here on the bottom right hand corner of your screen here, if this plasma membrane can allow these solutes to pass through, in which direction will these solutes pass? From left to right, correct. All the way until when? When they're equal out. What does it mean when they equal out? What's equilibrium? Dynamic equilibrium. Very good. And at dynamic equilibrium, when you have 4 on either side of that plasma membrane, does it stop moving? No. It continues to have solutes going back and forth, but it just in equal numbers. So it still moves past, but there's no net movement, overall movement. So yeah, this is just showing no net movement. It still goes passing through the plasma membrane. All right, very good. So there's different types of passive transport: simple diffusion, facilitated diffusion, and osmosis. Okay, the simple diffusion, there's certain molecules that are able to pass through the plasma membrane, and usually these molecules are very small. They're usually nonpolar molecules, and these attributes make them be able to pass through the plasma membrane really easily, and so they don't need proteins to help them. So they have to be very small, like I said. Nonpolar. Why do they have to be nonpolar? Anybody? Being small makes sense, right? So they could squeeze through, right? But why does it have to be nonpolar? What is this over here? The plasma membrane, very good. And the plasma membrane, is it hydrophilic, hydrophobic? It has hydrophilic polar heads over here, right? On the outside parts, they're hydrophilic, and that makes sense because it's touching with the outside environment, which is usually water or some sort of liquid substance. How about the middle over here? It's hydrophobic fatty acid tails, right? And if it's hydrophobic, can polar molecules pass through that hydrophobic layer? No. And that's why nonpolar or hydrophobic molecules are able to pass through, because it's like and like. Although, yeah, you might say, but Anson, over here they have polar heads, it's hydrophilic heads. How does it pass through? The amount over here is not that thick compared to the amount within the fatty acid tails over here. So it's able to pass through the plasma membrane freely if it's nonpolar and very small molecules. It has to be neutral also, which is kind of following that nonpolar aspect of it. So over here, it's basically the same animation where we had more concentration on one side, less concentration on the other. So it always goes from high to low concentration, and that's why it's moving to that location with lower concentration. Okay, for facilitated diffusion, it utilizes proteins to be able to transport those molecules over from high to low concentration. So these are usually molecules that are larger. So if it's larger, it needs the help of these proteins to be able to go through these proteins to the other side of the plasma membrane. It cannot go through the plasma membrane by itself. And these are the type of molecules that are polar as well. Since polar molecules cannot go through the plasma membrane due to the hydrophobic fatty acid tails of the plasma membrane, it utilizes these proteins to be able to pass through them. Same thing as charged ions and molecules as well. Same reasoning behind it. So over here, now you have this. You have this protein here that will help facilitate the fusion of these molecules. So that's if these molecules are too big to pass through, so it'll require this transport protein to help it through. So now it'll conformationally change. Okay, so this protein here, what is this area where it binds to this substrate called? No. What kind of site? Say again? No. Starts with the A. Active site, or binding site is also suitable as well. And this is the substrate, right? It binds to the active site. Causes a conformational change. What does conformational change mean? Conformational means, yeah, it will adjust, and then it will cause the enzyme to change its shape, conformationally change. Okay, so that's what it means. Conformational change means that it will change its shape. In this case, it changes its shape so that now it can release the substrate out to the lower concentration area. Okay. Osmosis is yet another type of diffusion. However, this is more specific to only water. So water will also go from high concentration of water to lower concentration of water. Okay, so it's kind of, you gotta think a little inversely a little bit. I kind of will give an example. Solutes, the definition, are the solids or the substances that end up dissolving in a solution or in a liquid. The solvent is the liquid portions of it. So in this case, we're talking about water. Okay, and that will end up forming the solution with the solute and solvent. It forms the solution. When we're talking about tonicity, we're talking about the concentration of that solution. So if it's isotonic, iso means the same. So it has the same tonicity or same concentration. And isotonic is when the outside environment has the same amount of concentration of solutes as the inside environment. So water will therefore, if you think about it, will be, there's no net movement of water. Some water will go into the cell, other water will come out of the cell because we have the same concentration inside and outside of the cell. So this causes the animal cells to be stable. And by the way, anybody can recognize this cell? What kind of cell is that? Red blood cell. Very good. Which a sickle cell is a red blood cell as well, but in this case, it's not sickle cell though. And for plant cells, the thing about isotonic water, if they're in an isotonic situation, it will actually cause it to wilt and be a little floppy. The reason behind it is because it actually needs to be in a hypotonic solution so that it ends up becoming a lot turgid, or it tends to be has a stronger it expands a little bit more, and that's what causes it to lift up. Okay, hypotonic solution. This is when the concentration of solutes inside the cell is actually greater than the outside. So meaning that water concentration inside is a greater or less. Yeah. Okay, concentration of solutes inside the cell is greater in this case. So the water concentration inside the cell, is it going to be greater or less then? less, right? If you have more solutes, you're going to have less solvent or liquid, or in this case, water. Does that make sense or no? I wish I had a... Hold on a second. Oh yeah, I forgot. I could just draw on here. Okay. So if we're saying this is the plasma membrane here, and we have solutes over here on this side, and we only have a couple solutes on this side. So if we're thinking about, and the water level is equal, okay, so here's the water level. I guess I should have drew that in blue color, but okay. So if you think about it, how much water is here versus here? Is there more or less on this side here? Less, right? Because there's more solutes here, making on this side more water. So in which direction will water go? Will it go to the right or left? Was there more or... Yes, you're right. So because there was more water on the right-hand side, so it will have to go to the left-hand side. Very good. Okay, that's if this membrane is impermeable or it cannot allow these solutes to pass through, but allows water to pass through. Versus, yeah, if it was permeable to these solutes, which direction will the solutes go? Right. Very good. The solutes will go to the right, so these solutes will go this direction if it was permeable to the these solutes. Okay. Okay, so in a hypotonic situation, since water will then go from the outside to the inside of the cell, it will cause animal cells to swell up, kind of like in this case scenario, and potentially lice as well. When I say lice, that will cause them to burst and break apart due to the amount of water pressure that accumulates. So in this case, it bursts like that. For animal cells, since they have a cell wall around their cells, and this cell wall will prevent them from bursting, this cell wall around the cells here, it will prevent it from bursting. So it actually makes them to be more turgid, and this is when it becomes more firmer, so that instead of wilting, this is where it tends to look like a very healthy plant. Hypertonic situation, this is when we actually have solutes that are greater in concentration on the outside. So there's a lot of solutes on the outside, not that much on the inside. So water concentration wise, which side is greater, inside or out then? The inside of water concentration is greater. So in which direction now will water go? Yeah, it will go from inside of the cell to outside of the cell to kind of balance the amount of concentration of water and also the solutes concentration as well. Very good. So the thing about, if water is leaving the cell from going from inside to outside, then it's going to cause the cell to shrivel up. So in both cases, both animals and plant cells, as you can see, it will cause the red blood cell here to shrivel up in this kind of situation. And also the cell inside a plant cell will also shrivel up. And so you see some of these in here. So this is in a hypertonic situation. Okay, so far so good? So this is kind of reiterating this. Looking at this, if this plasma membrane is impermeable to these green solute circles, these little things are water molecules. So yeah, it's the two hydrogen that makes up the Mickey Mouse ears, and then the oxygen that makes its face. But anyway, okay, if it's impermeable to solutes and permeable to water, in which direction will water go in this case scenario? Yes, water will go from the right to the left. Very good. Okay, let's say that this was the inside of the cell, and this is the outside of the cell. So what kind of tonicity is this solution that the cell is inside? Hypotonic, very good. Hypotonic because the solutes on the outside is not as much as inside the cell. Very good. And like we said, these water molecules will go from the outside to the inside of the cell. Very good. Okay. So now we kind of have it equaled. And once we have it equaled, now we have dynamic equilibrium, so we end up having it going back and forth. Okay, so same case scenario. Over here, what kind of solution are we in? Isotonic. So let's just say this side here is inside and this side is outside. Isotonic because we have it equal solutes on either side of the plasma membrane. And so in this case scenario, water will pass through these aquaporins and allow water to pass through the plasma membrane. Once again, we need these proteins, right, for water to pass through because water is polar. Very good. Okay, in a hypotonic situation, we have fewer solutes on the outside. And in which direction will water go? From in to out. Very good. And what will happen to the cell? Oops. So, actually from out to in. Do you know why? Okay, there's more substrates on the inside, right? How much water is in the inside in comparison? Not as much, right? Because there's more solutes inside than outside. So it doesn't have as much water molecules. While on the outside here, there's a lot more. There's a lot less solutes, but a lot more water in this case. I didn't draw enough water molecules in this case here, but you have to just picture it in your head. If there were less solutes on the outside, there should be more water on the outside then. So in this case, water will go from out to in, right? Because there's more water molecule on the outside. So graphically speaking, I didn't make it realistic. I should have put in extra water molecules over here on the outside to balance it out. Very good. So it goes in. In a hypertonic situation, hypertonic situation, it goes from in to out. Very good. Because there's more water molecules inside than outside. Okay, so what will happen to the cell then if this occurs? If it was in a hypertonic? It'll shrivel up. Very good. Because water from the inside will go out, causing the cell to shrivel up inside. Does that make sense? Yeah? Okay, now moving on to the next topic about active transport. So active transport, unlike passive transport, uses ATP. So we actually use ATP to cause the transport of molecules. One type of active transport are these protein pumps. And these protein pumps, like the name implies, it carries protein. These are proteins that pump molecules through them. So these molecules always go from now low to high concentration. And that kind of makes sense, right? Because if it doesn't use ATP when it goes from high to low concentration because it goes with the flow. But if you're trying to climb uphill or swim upstream, you have to take a lot more energy into doing it. So in this case scenario, from low to high concentration, it will use up ATP to be able to power that process. So this is going from low concentration, right, to high concentration. So it actually uses up ATP. ATP, when it hydrolyzes, it'll become what? Do you guys remember? If ATP hydrolyzes meaning that you use water to cause it to break apart a bond. Instead of adenosine triphosphate, it becomes adenosine diphosphate, ADP, and an inorganic phosphate with it. Very good. So once again, that it causes conformational change of the protein pump and exits the particular ion there, or whatever it might be. So the sodium-potassium pump, this is an example of this protein pump. It always pumps 3 sodiums out and 2 potassiums in, and it uses up ATP. This is kind of important because normally in a cell, there's more sodium on the outside and more potassium on the inside. So that's why we always want to have more sodium on the outside, and so pump these 3 sodiums out, but cause potassium to come into the cell. Just think about it for now. When we talk about the nervous system, it will make a little bit more sense. Exocytosis, endocytosis, all require ATP to cause it to work, and we'll go into each one of those. Okay. So this is an example of the sodium-potassium pump. Just looking at it itself, I guess you can't tell, but the red are the sodium ions, and the yellow here are the potassium ions. Okay, so the sodium ions, and this is going to be inside, and this is outside. The sodium ions tends to be more or less on the inside. Sodium ion. There's more on the outside, right, than inside. Potassium tends to be more inside than the outside. So this is normal. And in which direction, if we're going to use this pump, sodium will be going from in to out. We're trying to pump more out to keep the plasma membrane balanced, the membrane potential, but don't worry about that right now. So we're pumping 3 sodium ions out, and this is not going from high to low, right? We're going from low to high, so it will require what then? ATP. So we have ATP, hydrolyzes into ADP and inorganic phosphate. That will cause the conformational change of this pump, sodium-potassium pump, and then releases those 3 sodium outside. Okay, so we released it out on the outside. Now we're going to take in potassium, 2 potassium, and then the inorganic phosphate will dissociate, causing a conformational change of the sodium-potassium pump, allowing that potassium to now go into the cell. Okay, so that's the process of how the sodium-potassium pump works. Okay, exocytosis. So with exocytosis, what that happens is that there's these vesicles, which are these membrane-bound sacs. Okay, and these membrane-bound sacs will end up fusing with the plasma membrane, releasing whatever its contents inside out into the environment. So over here, usually it's docked over here next to the plasma membrane. So this is a vesicle, and there's something inside of it. If we're talking about the nervous system, the neuron will have these neurotransmitters inside these vesicles. And then when it's ready to send a signal out to the next neuron or to the next cell, it will fuse with the plasma membrane. And once it's fused, it creates this opening, causing whatever it contains within, if it's neurotransmitter, to expel into the environment outside. Endocytosis is basically almost the same thing, but the reverse process of exocytosis. It's actually causing it's engulfing something. So phagocytosis is a specific type of endocytosis. This is literally meaning that it's eating, cell eating. Phago means eating. Cytosis, cyte is cell. So how it does this, it extends its plasma membrane out, and then kind of encapsulates it and traps it in, and then brings it into the cell itself. Pinocytosis. Pino means drink, so this is cell drinking. And how this occurs, it's going to cause an invagination of the plasma membrane, and then the environment is liquid. So once it causes that invagination, it's going to just here's the plasma membrane regularly, if it's going to undergo penocytosis, it's going to cause an invagination of it. So whatever was out here is now going to be inside here, and then it's going to make a form a vesicle. Receptor-mediated endocytosis, very similar to penocytosis, except that it's even more specific in what's going to engulf. For penocytosis, it engulfs whatever is in that particular area. For receptor-mediated endocytosis, it needs to first have specific type of molecules that will bind to receptors on that membrane. And once that molecule binds to those receptors, that's when it's going to cause that invagination to occur. And once that invagination occurs, then we form that vesicle, which brings the vesicle or whatever substance from the outside into the cell. Okay, all this requires what? ATP, yes. Okay, so we did talk about, we're changing subject to talk about cell signaling. We talked about the receptor-mediated endocytosis. So there's these receptors on the plasma membrane that binds to a specific ligand. So once that ligand binds to, so in this particular case here, we have epinephrine as being the ligand. Here's the receptor here on the plasma membrane. Once epinephrine binds to this, I can't think of the word now, but binds to this receptor here, adrenal receptor, that's going to cause the signal transduction pathway to occur. And basically what that is this cascade of effects that will occur inside of the cell. So once it binds to it, it's going to cause activation of proteins inside of the cell. That's going to cause other proteins to be activated, so on and so forth, to finally cause some sort of response. In this scenario, when epinephrine does bind to it, when you think of epinephrine, it's adrenaline, so you'd be utilizing more glucose, and so it's going to try to break down glycogen into glucose molecules for us to utilize. Okay, and this is, you don't have to write down, just look at it, because this is very complex, but it's just to show you that signal transduction pathway. So over here is a granulosa cell, which is found in the ovaries of females, and on top here we have this receptor here, is the FSH receptor, and when FSH, which is a hormone, binds to this receptor, it's going to cause activation of proteins right next to it. So it first activates this G protein here. It's going to then activate the adenocyclase next to it. The adenocyclase will convert ATP to cyclic AMP, and this cyclic AMP will then cause other effects to occur. It's going to cause activation of this protein kinase A, which then does several different things. So when I talk about cascade effect, I mean this effect, this one thing, this FSH binding to the receptor will cause eventually several things to happen in the cell. Okay, so all this is occurring all because of this hormone binding to a receptor. And so this is the whole signal transduction. Do you have to memorize this? No. It would be terrible. Okay, I'm going to skip the question. We'll do it at some other time, but we'll go on and to the next slide. Oops. Pictures.